Lesson number 7. Pathogens are dangerous because they're constantly evolving. A slow evolving pathogen is not much of a danger because we can develop immunity to it and we can develop long-lasting vaccines to it. Smallpox and polio are very good examples of viruses that do not seem to evolve quickly. Therefore, smallpox has been pretty much eradicated. Polio pretty much gone too, where people are willing to take the polio vaccine. And that's something that I'll get to in just a second. But my favorite example, not my favorite actually, of a rapidly evolving pathogen is the flu. And in fact, if you're interested in something a little bit on the goofy side, search for my name and the Colbert Report. Because I think it's in 2006, I made 2 appearances on the Colbert Report. And in one of them, he asked me to prove that evolution was real. And I said the very fact that you need a flu shot proves that evolution is real. Because if you didn't believe in evolution, you shouldn't feel the need to get a flu shot for this year's strain of flu. Because if you don't think the flu can evolve, last year's shot would be fine. But good luck with that. This is a very primitive cartoon of what the flu virus actually looks like. There are 2 surface proteins exposed on the flu virus that are critical to its function. One is called hemagglutinin. Hemagglutinin basically binds to receptors on our own cell surfaces. So hemagglutinin is the key that gets into the lock of proteins in human cells. So that's how it gets in. So it tricks the cells into literally taking the virus inside. The virus uncoats and inserts its RNA genome into the cytoplasm. The other important protein on the surface is known as neuraminidase. Now, neuraminidase doesn't function to get into the cell, but when the virus replicates inside the cell and produces new virus particles, neuraminidase is important for the new virus particles to get out of the cell. So both of these are important functions. So it helps the newly formed virus particles leave the host cell. The viral genome itself is 10 strands of RNA. It's not a DNA virus, it's an RNA virus. So what happens is this RNA gets inside the cell. Our own enzymes basically, also brings in, I should say, an enzyme called reverse transcriptase that basically produces DNA, which can then be transcribed for the viral genome. So how do flu viruses make us sick? This is an illustration I copied from the New York Times. First, hemagglutinin proteins attach to the cell. That allows the viral RNA to enter. Then, once it's inside the cell, the viral RNA replicates. It produces messenger RNA for new code proteins to the virus. These particles then assemble, and virus particles enter via the action of neuraminidase. So that our cells are not only basically rendered non-functional by the virus, which causes the symptoms of flu, but our cells end up producing new virus particles, which is why the disease spreads so effectively. Now, it turns out there's 16 known forms of hemagglutinin and 9 known forms of neuraminidase. You're probably aware of this, or this will sound very familiar to you. Here is a chart showing basically combinations of hemagglutinin and neuraminidase subgroups. And it turns out these differ from one type of flu to another. There was a big so-called swine flu epidemic in 2009. That was an H1N1. Bird flu, 2011, H5N1. Avian flu, H7N9. And it turns out this year, there's an H1N1 floating around and an H3N2. The H3N2 turns out to be the more virulent form. I have asthma, a respiratory condition, so I am always eager to get a flu shot. And I'm there first thing. I do not want to get the flu with my breathing already compromised. And it turns out last year's vaccine was okay against the H1N1 version, but not so good against the H3N2. So as a result, I wash my hands constantly and do everything else I possibly can to stay away from the flu. But this is basically the problem. So these are viral surface proteins. They act as antigens to trigger the immune response. So when you get the flu, you actually do build up antibodies against it. And those antibodies basically are triggered against those 2 proteins. So viruses themselves are under selective pressure to form new combinations every year that our immune system doesn't recognize. And that means viruses are kind of locked into an evolutionary arms race with our own immune system. So once again, evolution is the driver here. Now, every year there are what you might call nuisance strains of flu that result from minor changes in the H1 version or the H3 version. This is called antigenic drift. They're annoying, they make you a little sick, but they're usually not life-threatening. Major genetic changes, however, can produce what's known as an antigenic shift. And these produce serious epidemics. Example, the pandemic of 1918. This is the great plague that I was talking about in recent American history. There's a wonderful American Experience program on PBS about the pandemic of 1918. It's chilling. This is a ward in a military hospital with young soldiers sick from the flu. This was all around the world. There were 50 million deaths worldwide in 1918 and 1919. That's a higher toll than World War I itself. And again, it was the conditions of the war that actually enabled it to spread. Almost 750,000 deaths in the United States. Think of that, 675,000 people in a much smaller population in this country, only about 120 million Americans at that time. So it was basically 1 American out of every 200 died in that year from the flu. I look back at my own grandparents and think, thank goodness you made it through. This was an extraordinary event. Could this happen again? Well, the same zoonosis that I've been talking about that were responsible for these epidemics are in fact still in action. And the reservoirs, especially for the flu, include birds, they include pigs. Horses are not up there for the flu. I put them up because of Triple E, but believe it or not, aquatic mammals, dolphins actually turn out to be a reservoir for some forms of the flu virus. But here's the issue. If genetic variation evolves, let's say, in a flu virus that's found in chickens or ducks, why is that a big deal to us people? And it turns out it's a big deal to us because of something we might call viral sex. And remember, one of my rules for how to excite students is about sex, okay? Well, viruses are very sexy. And I want you to imagine that flu strain number 1 and flu strain number 2 with different genes are both infecting a host cell. So they get inside, the viruses uncoat, all the RNA strands replicate, new viruses are assembled, and that assembly can have a random mixture of the genes from both of those viruses. This is as efficient as sexual recombination with meiosis and fertilization. And it turns out that a whole variety of new virions can pop out of the cell with new combinations of genes that basically now present a threat to the immune system of the host organism. So many of these viruses carry new combinations from both parental strains. So therefore, infections of multiple strains in any animal can result in new forms of the flu virus emerging. And that's the problem. Because these new combinations are sometimes very good for the virus in infecting and inverting the immune system, but very, very bad for us. For example, the bird flu that went around in 2013 was produced by antigenic shifts, viral sex, involving at least 4 different strains of influenza. And you can see the 4 strains right here. They were all avian, some of them in chickens, some in ducks, and some in pigeons, as it turns out. But they managed to mix together and then produce a novel strain, which was 8.7. I forget what the N was. Obviously, that's a typo, and I apologize for that. I'll go back and check. I think it was N9, but I'll double check that. But basically, a lot of our own serious problems were formed by that. So if you have viral sex, and you have strains that involved in different parts of the zoonosis, we could be in serious trouble. So steps to a possible flu pandemic are always there every year. So for example, you could have a chicken virus that somehow gets into a pig, is mixed with those, this is where swine flu comes from, and then could get into humans. You could have 2 versions of a virus infecting a human being and producing new combinations that infect other humans, or there could be a series of mutations that produce this. The replication of RNA in these viruses is very mistake prone, which means there's a very, very high rate of mutation, therefore there's a high rate of novelty.